I'm head of human genetics at the Wellcome Sanger Institute, and I chair the UK Biobank expert working group on genome sequencing and on the research analysis platform. So in this session, we're going to have a deeper dive into the large-scale molecular measurements of the biosamples provided by UK Biobank participants. And these are large-scale both in terms of the number of individuals being profiled, but also the number of molecular species being assayed in parallel. And these again terms such as transcriptomics, proteomics, lipidomics, and metabolomics. And so collectively, they're called multi-omics, and hence the title of this session. We have three great speakers today, and we're going to kick off with a discussion of the genetic data. As we heard in the first session on COVID-19 analyses, there's real challenges in disentangling associations for understanding what are consequences versus what are risk factors. And the genetic data can provide us with a really important causal anchor for many of these analyses. And we heard about techniques such as Mendelian randomization. It's also been hugely important as a kind of foundation stone of the UK Biobank resource, as we heard in the second session about the data analysis. So the three speakers that we have lined up I'll introduce them and also our distinguished panelists, and then we'll have the three talks back to back, and then the questions and the answers for all of those talks at the end with the panel. So our first speaker is Cecilia Lindgren. Cecilia is a senior group leader at the Big Data Institute at the University of Oxford. She's also co-chair of the International Common Disease Alliance. Her research interests are focusing on applying genetics and genomics for understanding the etiology of common complex traits, with a particular focus on metabolic and endocrine disorders. Our second talk on proteomics is from Chris Whelan. Chris is Associate Director and Head of Translational Genetics at Biogen, a pharmaceutical company, and his team focuses on the discovery of novel drug targets and biomarkers for neurodegenerative illnesses. And then our third talk, changing gears again, is from Professor Nilesh Samani. And Nilesh is Professor of Cardiology at the University of Leicester and a consultant cardiologist. He's Medical Director of the British Heart Foundation, and his research interests are in understanding the genetic basis of common cardiovascular diseases. And then joining those three distinguished speakers on the panel will be Professor Tony Whetton, who is Professor of Cancer Cell Biology at the University of Manchester and Director of the Stoller Biomarker Discovery Centre, where he uses proteomics and informatics to define disease mechanisms and biomarkers, especially those associated with cancer. So without further ado, I'll hand over to Cecilia to tell us about the genetics at a population scale and building maps for translation into mechanisms of medicine. Over to you, Cecilia.
Thank you, Matt. I'm thrilled to be here at the annual UK Biobank conference and to discuss the genetic resources in UK Biobank and how we are building genetic maps for translation into mechanisms and medicine. The remit I was given was to give a background in genetics, present the central dogma of molecular biology, building maps of common diseases, the UK Biobank genetic resources, and how it's useful for translation into clinical utility. So I will cram all of this into the next 15 minutes, which is quite the challenge, and I'm excited. So I'm going to start from the beginning here. The human genome consists of 3 billion bases, which are packaged into 23 pairs of chromosomes. In the genomic sequence, a range of elements or genes with exons that are the protein coding sequence are intercepted by introns, the non-coding sequence, and the genes are surrounded by non-coding sequences that hold promoters and long-range regulatory elements and sequences. The DNA is next transcribed into RNA, which is then translated into proteins. This is the central dogma of molecular biology and was first put forward by Crick in 1957 and published in 1958. It describes the flow of information in biological systems stemming from the DNA over to the protein. The black arrows you see here are observed processes, where the red arrows are not observed. And as Crick postulated, once the information has gotten into the protein, it can't get out again. So in the interest of time, I will take a leap forward here. There was a literal explosion in the genetic data field in the early 2000s. It's largely been driven by foundational data sets that together lead to the advent of genome-wide association studies. The Human Genome Project was launched almost exactly 20 years ago, or finished almost exactly 20 years ago a day. And it's a project that read out the sequence of the 3 billion base pairs in the human genome. And this monumental project led to the SNP consortium, which next provided the blueprint of the human sequence and the first glance of its diversity in terms of variation. A key insight that sprung out of these two projects was that when the genome was thoroughly examined, there is something called the human haplotype structure for common variants. It's relatively simple and sits in blocks of linkage disequilibrium, or haplotypes. This in turn led to the launch of the International HapMap project, which delivered insights into the common patterns of DNA sequence variation in the human genome. This information that markers travel together lent itself to the design of genotyping arrays, where you can genotype a couple of 100,000 variants on a chip or an array, and then use the knowledge of the underlying haplotype structure and linkage disequilibrium between markers to infer or impute nearby markers. So paralleling marker genotyping in this way drove down the cost, and suddenly thousands, and today even millions of individuals can be genotyped for a disease. However, even with that, and having delivered more than 100,000 robust association with hundreds of human diseases, even thousands of diseases and traits today, from the first robust findings, delivering the complement system robustly associated in age-degenerated macular disease by Klein et al. In Science, till today with the wonderful EBI resource catalog. And here I want to take a break and just congratulate Helen Parkinson and the EBI colleagues for that wonderful open resource. We note that we have been massively successful in mapping out these genetic association. A lot of people were very skeptical towards GWASs and what they would really deliver. I think it has been an absolute treasure trove for biological discovery. It has highlighted the role of microglia in Alzheimer's disease, sarcomeres in atrial fibrillation. It has pointed us to synaptic prunings being important in schizophrenia and autophagy in IBD. However, we can't rest on our laurels here. We're still left with thousands of diseases and traits that remain unmapped. Most of these associations are in the non-coding sequence, and they are common with a minor allele frequency over 1%. 80% of our genotyped samples are still of European ancestry, so we are lacking diversity, and we need to turn to sequencing to better understand structural variation and rare variation, and we need to do this at scale. So if we think that we're going to sequence, we have two options, and I'm just going to give you a quick overview of whole genome and whole exome sequencing. Whole genome sequencing doesn't require any selection of the areas you want to look, but as it implies, cover all of the coding and the non-coding regions in the genome, bar some repetitive sequences that are hard to sequence, and we're going to talk about those later, I think, in this session. It allows calling and cover all kinds of variants, including indels and structural variants that have been notoriously hard to call out of genotyping data. And as I mentioned, 90% of the common disease associations are in the non-coding regions. So whole genome sequencing really holds the promise of teasing out the full genomic architecture of common disease. While we think about that, though, we shouldn't ignore the whole exome sequencing, which is a lot of value for money. You focus roughly on 2% of the genome, only focusing on the protein coding part of the genome. It's cheaper, it's faster, and it's easier to analyze. A lot of us who are interested in drug targets and functional assays will also know that the coding variants, at least from what we can see so far, seem to have larger effect sizes and therefore be easier to sort of assay functionally, but that remains to be proven. So what has been done in the field so far then? The first effort showing its scale how low frequency rare variation mapped out in the genome was the Exome Sequencing Aggregation Consortia, or EXAC, led by Johnny MacArthur and colleagues. Exome sequence at scale were jointly called and analyzed. This has later been preceded by the NOMAD effort, which currently holds 15,000 whole genomes and about 125,000 exomes. it does point us to a high burden of protein truncating variants, or predicted loss of function variants, as they're popularly called. And I note the predicted here because before variants actually have been shown to have a function, we rely on predictions. The data also holds an enrichment of disease samples, and it doesn't have a lot of phenotypes available. It's been an absolute treasure trove again for the field to benchmark exome sequences, especially in rare disorders, to see if the variants you're looking at are really rare in the wider population, or if it's a sequencing issue in your own or opportunity in your own data. It, however, still leaves disease researchers in need for large-scale, well-powered, deeply sequenced samples where they also have a rich tapestry of phenotypes available. And where better to do that than in UK Biobank? So if we move forward then, UK Biobank has been genotyped and has been imputed. So for instance, if you use the HRC imputation, you will have over 28 million markers passing UK and QC, and you will have about 8 million markers that have a minor allele frequency of less than 1%. So why do we want to sequence them? So there's been a number of papers showing exciting phenome-wide analyses of these rare variants and showed genes with strong pleiotropic effect, associations with a range of diseases, as well as non-additive phenotypic effects, adding to what we would typically find in a GWAS. However, as been discussed widely, both on Twitter and in the literature, genotyping chips and imputation are less reliable at lower allele frequencies, particularly less than 0.1%, than sequencing. And it only assays at already known sites. So any novel variants, private variants, or variants with unstable QC will be missed or misrepresented. So the release of new sequencing data will fill in this in the sequences. So as you will have heard previously today, but from Jeff Reed and Mark Effington, the UK Biobank whole exome sequencing efforts is currently ongoing. It's a public-private partnership led by Regeneron. It released its initial 50,000 samples in March 2019. A subsequent release of an additional 150K exomes, taking it to 200,000 samples in total, were made late 2020. All of us are working on these samples at Anger, and we're anticipating the release of a further 100,000 exomes, taking it up to 300 in total mid-2021, and the full release should follow quite shortly after, with an estimate about for end of this year for delivery. So searching about this on medRxiveX and bioRxiveX, I found over 180 manuscripts on medRxiveX preprint service to date on UK Biobank exome sequencing. And I want to point out that Dr. David Curtis in London holds the record on 23 papers as a single author on these data. So looking at what it has really delivered then, the first tranche of exome sequencing, it does remind us that quality control and quality assurance is the individual responsibility of anyone working with this data. It's been a wonderful learning curve for a lot of us. 4 million coding variants were delivered, and almost 98% had a minor allele frequency less than 1%. It delivered almost 200,000 predicted loss of function variants, and that's a 14-fold increase compared to the imputed and genotype data. They also show that concordance is shaky for variants with a minor allele frequency less than 1% in the genotype data and also in the imputed data. The data aligns very well compared with the non-Finnish European ancestry samples in NOMAD as a quality control. And of interest to those of you who work in the rare disease field, 2% of individuals carry a pathogenic variant in at least one of the 59 genes that the American College of Medical Genetics and Genomics have compiled, for which mutations are known to be causative of various disorders and clinically actionable. So obviously, already with 50,000 genomes, quite a lot of knowledge was put forward. That has sort of been taken on now, and the second tranche holds around 200,000 exome sequences. Now also a large number of traits have been put forward by a number of groups, tens of thousands diseases and over 1,400 quantitative traits have been put forward. Almost 1,000 novel associations have been put forward here. One of the things that struck me and that we have discussed previously, but was shown here for the first time at scale, is whether these low-frequency coding variants would really display larger odds ratios, which one would expect. And there is a median odds ratio of over 15 for the analyzed diseases. Previously common variants, when we looked at gene-based tests or collapsing methods, have barely delivered anything that wasn't picked up on single variant tests. But here in the exome sequencing data, about 80% of findings were only seen when we were looking at gene-based data, especially pointing to the allelic heterogeneity here. It is also noteworthy that increasing the sample size 3 to 4 times lead to a 12-fold increase in study-wide significant association. So the incoming tranche of exome sequencing data will further propel the discovery of associated coding variants in European descent samples, which is an amazing resource for functional follow-up and translational effort. However, I want to emphasize something that my colleague and peer Gonzalo Abecassis mentioned in a talk yesterday, that if we're serious about alleviating disease burden globally in a more equitable way, we need to expand our efforts into other geographic regions. I want to point out that UK Biobank is not purely a European descent sample, but holds other ethnicities as well. And there are ongoing global efforts in ICDA and so forth that I will talk about later, and we've seen wonderful papers from TopMed, Gene and Health Cohorts, Qatar, Africa, China, India, and Japan, so forth. So it's something that the field is rallying behind and pushing forward. So there is also an ongoing whole genome sequencing effort in UK Biobank. And again, it's a private-public partnership where about 200,000 whole genomes are being sequenced and should be publicly available sometimes in the fourth quarter of 2021, which is amazing. The sequencing project itself is progressing well despite the pandemic, and it's more than halfway complete. We'd anticipate that the full sequencing should be complete sort of sometime at the end of this year, with data available for all 500,000 samples in early 2023. And again, it's an amazing effort that will further diversify the allele spectrum covered and deliver a finer map of associated diseases and traits, again pointing out that the non-coding genes are expected to hold about 90% of the disease associations. So with this in mind, now sitting with a map, a genetic map of thousands of associations, both in the coding regions and the non-coding regions, we are powering on with putting a lens on the next bottleneck, and we face a big challenge here. So how do we move from genetic risk variant to their function? How do genetic risk variants elicit the function on a physiological system? How do we find the causal variants for each association and locus, and how do we make sense of these? There are a couple of key questions that we need to ask ourselves. What are the target genes or effector genes through which these associations exert their effect and elicit phenotype? For many diseases, we don't even know the full picture of target tissue or target cells, which is absolutely crucial to be able to do proper functional work. What is the function of the effector genes, and what's their correlating proteins? Chris will talk about the proteins later, but even in the widest arrays, we don't assay all of the proteins yet. What are the pathways involved? We don't have a full pathway map of the entire human genome. And these are something we sort of really need to question ourselves and think about how we should sort of propel forward. We also should be curious about developmental time points that are crucial for developing disease, both on an organ level, an organism level, and on a cellular level. And pushing forward with this, we have come together in the field, and we have thought about very deeply how to rapidly move from discovering loci of disease-associated variants to understand disease mechanisms and propel therapeutic development. We must develop systematic ways to discover and accelerate all of these steps to improve research, to increase efficiency and innovation into clinical translation, and most importantly, improve care to address these questions. And we should also do this in a globally equitable way. So this has led over 150 scientists in the world to come together to think and formulate the Maps to Mechanisms to Medicines challenge. And this discussion has also led to the foundation and the launch of the International Common Disease Alliance, which serves as a scientific forum comprising of international stakeholders across academia, medicine, biopharma, tech companies, and funders that really sort of come together to tackle these questions and think about a foundational data set such as UK Biobank that needs to be stood up for us to be taken seriously and to solve these issues. So in conclusion, we are at the pivotal moment in biomedical research with a width and a depth of data never before available. It's breathtaking and it's super exciting. We have over 100,000 robust genetic association with disease delivered. The whole genome sequencing and the whole exome sequencing in UK Biobank offers unique opportunity to expand this several fold through exploration of the rare allele frequency spectrum at scale. The remaining challenge is for translational impact, how to propel ourselves from genetic discovery into disease mechanisms. And I want to point out something here that this also serves as a test bed for how effective and successful public-private partnership can be. So with that, I want to thank everyone who's involved in the UK Biobank, the funders, and of course, all of the UK Biobank participants without whom none of this could be done. Thank you very much.
Thank you very much, Cecilia, for a lovely exposition there of all the genetic data. But just to remind everyone, we'll be taking questions at the end of the three talks, so please put your questions into the Q&A and we'll follow them up later. So our next talk is from Dr. Chris Whelan, and he's going to be talking to us about proteomics. So over to you, Chris.
Thank you, Matt. Yeah, it's a huge honor to speak at today's conference. My name is Chris Whelan. I'm a scientist at Biogen in Cambridge, Massachusetts, and I'm going to speak about the Pharma Proteomics project. This is an effort to perform blood biomarker profiling on 56,000 samples from UK Biobank, pushing forward what we hope will be a new era of population-scale proteomics. Now, when I say proteomics, of course, I'm referring to the study of proteins. Proteins are the building blocks of life. They have countless functions in the human body, and they represent the vast majority of drug targets. In other words, they're the most common molecules against which we develop new medicines. And thousands of proteins can appear in blood, and that can potentially provide us with a window into human health. So, for example, certain versions of a protein called tau can act as powerful early indicators of Alzheimer's disease, whereas another lipid-based protein called ApoB is an established blood marker for hypercholesterolemia. And typically, these kinds of powerful protein biomarkers are discovered using hypothesis-based trial and error kinds of experiments. But very recent advances are starting to facilitate more unbiased discoveries. And so we're now entering an era whereby proteomics can sit alongside genomics and transcriptomics as a technology that can be applied at a population scale. So let's talk about this technology in a little more detail. Broadly speaking, you can apply two different kinds of proteomics. On the left of the screen, you see that there's a targeted approach whereby you take your patient's sample, let's just assume that it's a blood sample here, and you feed it into an immunoassay that is specifically designed to detect usually a single protein of interest. So this is an excellent technique. If you have a single protein in mind, it usually detects that protein with very high accuracy, but of course, it's less useful if you want to perform unbiased discovery. And so that brings us to multiplex proteomics. Multiplex proteomics is a way of detecting many different proteins at the same time. And so on the right of the screen, the oldest and probably most widely applied kind of multiplex proteomics is mass spectrometry. And that measures the mass to charge ratio of ions in a patient sample. It's arguably the most unbiased way of performing multiplex proteomics, but it also requires very large sample volumes. It sometimes misses lower expressing proteins such as inflammatory cytokines. And perhaps most crucially, it's difficult to scale up to a very large data set like UK Biobank. So that brings us to the middle of the screen, to what you might call the Goldilocks proteomics method, or more formally known as affinity-based proteomics. And so with affinity-based techniques, much like mass spec, they can detect hundreds, if not thousands, of proteins simultaneously. But the difference here is that these techniques rely on a backbone library consisting either of aptamers or antibodies designed to capture specific proteins. And since these techniques rely upon such pre-selected libraries, they're not fully unbiased. But the huge advantage here is that they're very good at detecting lower expressing proteins, and they don't require higher sample volumes, and probably most importantly, they do scale to large data sets like UK Biobank. And needless to say, UK Biobank is unrivaled in the sheer breadth and depth of data that's already been collected. It's the perfect data set in which to employ proteomics at massive scale. As you've heard in Jeff's talk and Cecilia's talk, biopharma companies like Regeneron, Biogen, and many others have made substantial investments into the generation of gene sequencing data from UK Biobank. And the reason that we've made these investments is to help accelerate what we call genetics-guided drug discovery, as you can see on the right of my screen. So essentially from this graphic, what we do is we apply techniques like genome-wide association and rare variant burden testing to identify gene variants that are linked with increased or decreased risks of the diseases that we want to treat. And if we find a particularly compelling new gene variant, it might point us towards an important new protein involved in disease. And then we can target that protein with a new medicine. You'll notice, however, that a key component of this discovery process is currently missing: the protein targets themselves. So right now, we don't systematically integrate proteins into our genetics-guided drug discovery process. We largely assume that there's a direct link between gene variants and disease endpoints. But as any geneticist in the audience will tell you, that's often not the case. And that's what motivated our investment in the generation of proteomic data in UK Biobank. Measuring thousands of proteins simultaneously in the blood samples of UKB participants will allow us to bridge the gap between disease-associated gene variants and the diseases themselves, pointing us more explicitly towards drug targets. And our hope is that this will open up a whole new avenue for drug discovery, one that's guided not only by the blueprints of human diseases, in other words, our genomes, but also the circulating end products of those blueprints, in other words, proteins. Now, this is all quite exciting, but suffice to say, proteomics is still a relatively expensive technology. We're at a point with proteomics which is comparable to that of genomics a generation ago. It used to be prohibitively expensive until grants from excellent government bodies like the Wellcome Trust and the Medical Research Council came in and basically helped the technology become cheaper, more accessible, and more widely employed. And then it helped the technology follow and eventually surpass Moore's Law. Proteomics, much like genomics, could eventually be routinely employed as a healthcare tool. It could hold as much, if not more, transformative potential. But we really do need to all come together, academia and industry alike. And that is what motivated the formation of the UK Biobank Pharma Proteomics Project, or PPP for short. So PPP is a consortium of 10 companies. I'll take a big breath. Biogen, Amgen, AstraZeneca, BMS, Genentech, GSK, Janssen, Pfizer, Regeneron, and Takeda. So we are exclusively funding a generation of just under 1,500 new protein measurements from 53,000 people in UK Biobank. We plan to use these data to help develop new medicines and diagnostics. We will, of course, release all data to the broader research community after a short embargo period. And so to generate these data, we'll use an affinity-based multiplex proteomics technique developed by a Swedish company called Olink. Olink's technology was selected after nearly a year of due diligence, and many factors will influence our decision to move forward with Olink, including the quality of the platform itself, its pricing, its scalability, the turnaround time, and perhaps most crucially, the company's ability to contract with 10 major biopharmaceutical organizations under a very accelerated timeline. We needed to get this done basically and wrapped up in 2020. Now, Olink's platform currently measures just under 1,500 proteins, but that number is actually expected to double in the third quarter of this year, and we're currently exploring ways to fund expansion to those full 3,000 proteins, so stay tuned on that. Now, as I mentioned, this is a slightly technical slide. I may lose some of you, but Olink's platform is affinity-based, so it relies upon, as you see in part A here, it relies upon masked pairs of antibodies carrying DNA oligonucleotide tags. And those tags are developed to detect unique proteins in a patient's fluid sample. When these matched tags are brought close to the protein they're supposed to detect, they undergo a process called hybridization. And that hybridization can then be used as a template for extension into what we call amplicons via polymerase chain reaction. And those amplicons essentially act like barcodes, and those barcodes can be easily read by a next-generation sequencing platform, such as Illumina's NovaSeq 6000. So I don't want to labor too much on these details, but suffice to say, the technique is it's a great combination of old and new, antibodies and next-generation sequencing. Moving on quickly, just suffice to say that we'll be applying Olink's technology to 53,000 UK Biobank participants, 56,000 plasma samples in total. And you might ask, why is there a discrepancy in the numbers here? Well, 3,000 of the 53,000 participants will come from an ongoing longitudinal COVID-19 imaging study, the same study that Paul Matthews alluded to this morning. Now, we plan to run Olink on all 3,000 study participants across 2 different MRI scanning sessions. So what we'll build here is the world's only investigation of the imaging and proteomic signature of COVID-19 pre- and post-infection. Beyond the 3,000 COVID study participants, will profile an additional 45,000 people that are selected at random in order to avoid enrichment and keep it as representative of the broader UKB population as possible. And then there'll be a final 5,000 samples that are cherry-picked by the different companies. Now, we expect PPP to spearhead many exciting new scientific discoveries. Notably, it will facilitate the largest ever study of genetic influences on protein concentrations. In other words, the largest ever PQTL discovery, GWAS. PQTLs, very quickly, are genetic loci or SNPs that are known to influence variation in protein concentrations. And we often use these PQTLs as instruments for a powerful mathematical technique called Mendelian randomization, which helps infer causal effects from genetic studies. And so you'll see in the bottom right of the screen, one recent example comes from the lab of Brent Richards at McGill University. Brent's team showed that a genetic variant associated with reduced COVID-19 severity was also associated with increased levels of an enzyme called OAS1 in lung. Kenneth Bailey spoke about this earlier today. And so Brent's team applied Mendelian randomization to indicate that OAS1 indeed has a causal role in COVID-19, potentially validating it as a new drug target. And that's an example for just one protein. We'll be generating data on 1,500. So beyond genetics and PQTLs, there's also huge potential to accelerate biomarker discovery and to improve our understanding of many complex diseases with these data. For example, we can use the Olink data to scrutinize how neurological markers like tau and neurofilament change in people that are at higher risk of Parkinson's or MS. We can explore more established markers of cancer and cardiovascular disease. And then going back to genetics, we can look at people who carry loss of function mutations in certain genes to determine how those loss of function mutations impact their proteomic signatures. That's just a handful of examples on how the data can be used. I'm hoping this talk will inspire dozens of other ideas. And maybe you already had an idea 10 minutes ago and you just want to tell me, you want me to tell you how to get the data and when the data will be delivered. Well, the good news is that the data will be delivered in quite a simple format. It'll be a CSV file, very easy to manipulate in your favorite tool like R or Python, or you could even use Excel, but as a data scientist myself, I'd rather that you didn't. But for anybody who is less computationally savvy, there is actually a completely free tool on Olink's website available to all researchers, which makes it very easy to manipulate these data. Just in terms of timelines, the first batch of plasma samples is expected to ship from UKB to Olink's Swedish facilities in 2 months' time, April 2021. New batches will be delivered every month until October, and then the pharma consortium will receive linker codes for 3 different batches of data, one in June, one in August, and one in October. All approved UKB researchers will receive their own copies of the data 9 months later, the first 18,000 around this time next year. And so with that, this is my final slide. I hope that I've emphasized how plasma proteomics will not only enhance the UK Biobank resource, but also accelerate biomarker discovery and drug development. We believe that this represents the first major step in what could be a much larger, more ambitious revolution for proteomics and precision medicine. It could potentially expand to cover more more proteins and eventually capture the entire UK Biobank population. But in order for that to happen, we really do need to come together, government, academia, and industry alike. And so Rory, Naomi, Matthew, and the entire UK Biobank team have already shown the world what wonders we can achieve through scientific collaboration. And I believe that the best is yet to come. I want to thank them, as well as the many, many, many pharma partners you see here, particularly Melissa Miller of Pfizer, Linda Mitnall of Regeneron, and the business development teams at AZ, GSK, and Amgen. And of course, all 500,000 participants in UK Biobank for making this possible. I'm hoping that we can come back and speak again next year in real life with real scientific results. There's a lot to look forward to, and thank you very much for listening.
Wonderful. Thank you very much, Chris. That was a very enthusing and motivating talk, and we very much look forward to next year and hearing about the results of the proteomic work. So our third talk in the session, and the last one before the panel, is from Professor Nilesh Somani. And Nilesh is going to be telling us about telomeres in UK Biobank. Over to you, Nilesh.
Thank you very much, Matt. It's a pleasure to join you and other colleagues. It's an honor to present this talk about our work in telomeres on behalf of my colleagues who worked on it. Just to set the context for this work, I want to this is something that I think a lot of people see when they look at patients with diseases. We know that many diseases are age-associated, but not an inevitable consequence of aging. So if you look at the graph of coronary artery disease, which is a typical age-associated disease, only about half the men get it, and only about a third of women get it. And therefore, the question is, why do some people develop this disease, and why do they develop very early, and other people with the same risk profile never develop it or develop it very late? There are, of course, many, many explanations for this, but one of the components is telomeres. And I think for those of you who are not familiar with what telomeres are, these are the ends of your chromosomes, and they consist of a sort of repeat sequence which caps the chromosomes, and their primary function is to protect the ends of chromosomes. And there are particular properties of telomeres which are relevant to this hypothesis around why they may be involved in age-associated diseases. So in this schematic, what's shown, we are all born with telomeres shown in dark blue here of varying sizes, okay? And as cells divide, for a number of reasons, telomeres shorten with cell division. And they have a fundamental role when they reach a critical small size to make the cells senescent. So this variation in telomere length that we are born with, the variation in how frequently they shorten, and the relationship with this cell senescence makes them a biomarker of what one might call biological aging. And just to give you an analogy of how this might be related to the development of diseases, if you believe that telomeres are this marker of biological aging, I would just sort of use the stars as the tire tread as an analogy. So we see this is a fresh star here on the left-hand side, showing deep telomere, deep tread. And if you can imagine that, depending on what you're born with in terms of the tread size and how this relates with wear, you could end up with tires that, of course, have very thin treads at this age. And this will vary from individual to individual, related to both what you're born with and the age-related attrition that may itself be related to both biological factors in individual and external lifestyle factors. So that's the sort of underpinning hypothesis about a relationship possibly between telomere length that you have and your risk of some of these age-associated diseases. We started this work 2 decades ago when we first started to look at the relationship between telomere length measured in blood samples or blood DNA and the risk of coronary artery disease, which comes from my clinical interest in cardiology. And I don't have there are lots of examples of this, but this is one of the papers we published some more than a decade ago, where we used a clinical trial called the West Coast Scotland study, where they took some baseline DNA and followed up people afterwards who were given or not given a statin to see whether they benefited from statins. And what we are able to show from baseline DNA that was collected is that if you had shorter telomeres, as shown in the if you look at the blue graph, then blue histograms, then you have your risk of developing coronary disease, allowing for other risk factors, was almost doubled in those people who had shorter telomeres compared to those with longer telomeres, as shown in the first tertile. And the statins tended to benefit particularly people who had shorter telomeres. We've gone on in other studies, as alluded to by the speaker, that there's of course a technique called Mendelian randomization, whereby you could use genetic information to understand whether the relationship between telomere length and the disease is causal. And in some work we published a few years ago, we mapped a number of genetic loci that affect your telomere length. It's a highly inherited condition, so about 70% of inter-individual variation in telomere length is genetic. We were able to map the first 7 or so loci for telomere length and use this in this approach called Mendelian randomization to see if genetically determined differences in telomere length related to coronary disease. And as you can see on the graph, which is a meta-analysis of a number of these studies, we see that indeed there is a relationship between telomere length, shorter telomere length, and higher risk of getting coronary artery disease. So this gave us the impetus to say, could we measure telomere length in UK Biobank? And UK Biobank, as has been alluded to in some of the other talks, clearly is a tremendous resource in which to integrate telomere length measurements, because of course there's a comprehensive curation of diseases and biological traits to robustly test for association with telomere length. The fact that there's extensive genomic and exposure information can also help us identify what are the determinants of telomere length. And we can compare, which has not been often been possible in many studies, we can compare genetic and observational association, same population, and make causal inferences. So a few years ago, we started this project about measuring leukocyte telomere length in UK Biobank. We were very fortunate because rather than array a new sample for telomere length measurements from the DNA, we were able to piggyback the fact that UK Biobank colleagues had arrayed the samples for the genome-wide association, the genotyping that they had done as part of the genotype measurements that they did in UK Biobank, which most of you are familiar with. So we were able to get the residual samples from the genetic analysis that was done, and then we measured telomere length using a PCR-based assay. And I just want to highlight colleagues who, this is an academic enterprise. This isn't one that was led by industry. This was in a university academic lab where we met the challenge of trying to measure UK Biobank in this very large sample set. Brian Cord, who is pointed out in the left side, is a leader for ensuring that the measurements were made, and Chris Nelson has been the senior statistician on this project. And I just want to highlight colleagues, 6 dedicated research assistants who over 4 years made these very large-scale measurements in UK Biobank. So we received just under 489,000, just over 489,000 samples. Some of them we had to withdraw, of course, because UK Biobank often asks us to withdraw a few individuals. And we were able to make measurements in 488,427 subjects. And we were able to get valid measurements, and we'll be publishing what we mean by valid measurements in due course, in over 97% of the samples that we received, which I think is a remarkable achievement. Most of the time, the reason we couldn't get a result was because there was insufficient DNA available from the sample we received. This is a project that's gone on, as I said, for over almost 4 years in terms of measurement. And of course, during that period, summer and winter comes. We have different primer sets. We had, a dozen or so PCR machines. We had 6 operators. We had 4 robots to array the samples. So we had to have very stringent checks on whether we maintained efficiency of the assay and made sure that we adjusted for everything. What is shown on this slide is that over 200 weeks of assaying, there was a fairly consistent relationship between weekly distribution of TS ratios across the whole duration of the study when we adjusted for technical variables. As I said earlier, we will be publishing this shortly. We also introduced within the sample, it says, of course, some duplicates to see how the assay performs over time, and to show that there wasn't a wide variation in results. And amongst over 22,000 samples, just under 5% of the samples repeated measurements throughout the course of history. We had a fairly reasonable coefficient of variation in this assay measurement of an average of just under 6%. UK Biobank also introduced some blinded samples, which gave a very similar coefficient of variation as well. So very reliable samples, we believe, measurements that we think we obtained. There are a number of well-established relationships between telomere length and certain characteristics, and we were able to reproduce this in this large study. So what's shown in the graph on the left-hand side is this well-known relationship between your age and then attrition in telomere length with age. So we saw that, and women shown in the sort of pinkish color, the male color, men in the blue, tend to have slightly longer telomeres, and we also were able to see this very clearly within this sample set. But what I wanted to remark on is the enormous inter-individual variation you see in telomere length between individuals at any age in the graph on the left-hand side. There are some very not well-reported differences in ethnicity in terms of average leukocyte telomere length, and we were able to show, as has been shown previously, that black subjects tend to have slightly longer telomeres than people who are white. We also shown now that Chinese people tend to have slightly longer telomere from the small sample set that's available within UK Biobank, as are people from mixed ancestries, and indeed South Asians. They tend to have slightly longer telomeres. Clearly, as you've heard from previous talks in this session, there is, of course, the opportunity of looking at the genetic determinants of telomere length, and we've done a last GEO study on this, and we have identified, after a lot of checks and so on, that we identified just under 200 conditionally independent variants at a higher stringency than normally adopted for GEO studies, which determine telomere length, which are in 138 loci, of which almost 80% are novel. And we supported this by further analysis using rare variants from the exome data that's available in UK Biobank, showing that many of the candidate genes at this loci are probably truly causal in this. So we use this information, of course, to then say, can we therefore study both the observational and the genetic association of telomere length with diseases? And we sort of curated both incident and prevalent cases of diseases across a whole spectrum of things in UK Biobank, about 123 diseases. We've looked at ranging from cancer to cardiovascular diseases to diseases of other sorts. And we have seen about 16 associations which genetically, after bone foreign correction for the number of tests we've done, are highly significantly associated with telomere length. And a further 15 diseases which show nominal genetic associations, so about 30 out of the 123 that we looked at, so some evidence. And in most cases, we see concordant observational associations as well with telomere length. And as you can see from the graph, it's a little bit difficult to read, and apologies, it's a busy slide which shows these 31 associations that I pointed out, that we see effects with both longer telomeres and shorter telomeres. So many cancers, the increased risk of cancer is associated with longer telomeres, whereas for many age-related non-cancer diseases, there tends to be an association with shorter telomeres. Just to illustrate again the relationship I already mentioned for coronary artery disease, because of our special interest in this, what's shown on this slide is the results for coronary disease. So as you can see, the observational association, which is based on incident diseases after their recruitment to UK Biobank, we see this significant increase with one standard deviation, shorter telomere. In the genetic association, which we use for prevalent and incident cases, we see a similar relationship. And what the graph on the right just shows that this is a linear relationship. There isn't a threshold where we see an effect. And we also looked, initial analysis about 100 biomedical traits, and we see relationships that are causal between very diverse biomedical phenotypes like height, associated with telomere length, to bone marrow function. But I wanted to end really by showing you something that has been a long debate in the literature around the telomere length, which is, does it impact on life expectancy. Obviously, there's a relationship with a whole bunch of diseases, as I've shown you just now. And that's it. If you aggregate all of those, is there a relationship between shorter telomeres and your average expected life expectancy or longevity? And that's been a really open question in the telomere world, telomere field. And of course, UK Biobank now provides us with a scale to look at this in detail. And what's shown in the slide is a modeling of this work. So for males and females separately, but very similar results in both genders, that compared to people who have the longest telomeres, which are shown right at the bottom, on average, people with shorter telomeres as you go beyond so standard deviations, if you take the people with the shortest telomeres, there's an average of somewhere around 2.5 years worth of average life expectancy lost or expected to be lost in people who have shorter telomeres, taking aggregate amounts of aggregate assessments of this. And when we split this into whether this is due to cardiovascular disease or cancers or non-cancer or non-cardiovascular diseases, all of those contribute to this, but particularly the sort of non-cancer, non-cardiovascular diseases. So this is not based primarily on the relationship between telomere length and cancer and cardiovascular diseases. So I hope I've given you a flavor. I mean, this is a big resource that we have developed. I hope I've shown you that we created really for the research community, the world's largest research to investigate the determinants and biomedical consequences of individual variation in telomere length. As I hope I've shown you, our initial analysis of this has demonstrated the widespread influence that this variation has both on traits and particularly on diseases, spanning multiple body systems and on overall life expectancy. I earlier showed you the colleagues who were really responsible for the generation of the telomere length data. These are colleagues on this slide between my team in Leicester and John Dinesh's team in Cambridge, who really spent the last year mainly on Zoom calls at least once a week, trying to start analyzing some of these data. So I want to really thank them and just acknowledge them for that work. And finally, of course, the measurements of telomere length in UK Biobank were funded by three particular funders: the MRC, BBSRC, and the British Heart Foundation. So thank you.
Wonderful. Thank you very much, Nilesh. That was a brilliant exposition of a brilliant data set. So we very much look forward to seeing those results. And now we move on to the panel session, and I introduced Tony Wetton a moment ago, and Tony, just to give you a little opportunity to comment on your perspective on the kind of the panoply of omics available within UK Biobank, both now and in the future, and what your thoughts are about that.
Okay, thanks. Well, they were three great presentations, and of course, Cecilia started with the DNA makes RNA makes protein dogma, which is something that the UK Biobank can integrate and study at great scale. And Chris gave a great presentation on the business of how we're going to measure proteins. And the scale is, of course, the thing that is so amazing about the UK Biobank, in the fact that we can integrate genomic, proteomic, metabolomic data. And I think there's one question come in about metabolomics. So in terms of the Nightingale metabolomic project, it is ongoing, and the first data release on 123,000 metabolomes is imminent. And there is actually a preprint on the data that they've generated concerning metabolomic data and risk of severe COVID-19 that's available, as I say, as a preprint. So we've got 56,000 samples going to be run with Olink. It's not going to stop there. There are going to be a whole variety of other assays take place with UK Biobank biofluids that are available, be it plasma, serum, or urine, saliva. And the amount of data that you can get from tiny quantities of material these days means that the resource is not going to be denuded for, I would say, decades. There's really a great deal can still be done. And that means in the future we will get greater detail on the metabolome, on the proteome, and in respect of the proteome, we'll be able to look at post-translational modifications and the like, and we'll be able to look at polymorphism, proteoform analysis, and go on a deeper dive into these various omics and relate them back to the nucleic acid-based omics. So the way that we'll take this forward is, of course, by considering the best technologies that are available. And indeed, we have a workshop on proteomics and how to develop proteomics in UK Biobank coming up later this year.
Brilliant. Thank you very much, Tony. It's a very exciting kind of prospect that you painted there. So moving on to the questions now, and thank you everyone for submitting your questions. There have been an awful lot of them, so I'm afraid we won't have a chance to go through all of them. But thank you. And maybe if I just start with one question that came in, which ties together the first two talks. So it would be good to get Cecilia and Chris's take on this, which is that Cecilia commented that many of the associations are expected to be in non-coding regions. What might an association between protein levels measured by Olink, as Chris described, and non-coding variants suggest about the underlying biology? And maybe, Cecilia, do you want to kind of comment on that first and then over to Chris?
Yes, so I think the variants can, as I think Chris very elegantly talked about, PQTLs, where you have a genetic variant that is associated with the level of protein that sort of comes from the gene that is encoded. And as I showed in one of the first graphs, if you have an association in one of the regulatory elements, you can affect the expression levels and hence the protein levels of the gene. And then the protein in turn would be expected to elicit their effect on the subsequent sort of physiological activity or disease or a phenotype. So that would probably be through amount, I would think, but there are other mechanisms too for that. Chris, do you want to comment as well?
Absolutely. No, I couldn't put it much better than you, Cecilia. Thanks. I think that this would actually be quite exciting, and what it might imply is that what we once used to call junk DNA actually might have a really crucial role in disease processes. It would indicate to me that trans PQTLs, in other words, variants that influence levels of proteins coded by far away gene regions, actually play a really crucial role in disease processes and are worthy of much further interrogation. Our hope is that we do find a lot of non-coding regions that influence these protein levels.
Yeah, and as I mentioned, 90% of patients are in those regions, so you would expect to see some of those.
Thank you both. And then to introduce another omics, and just in case people aren't confused enough, there's an interesting question here for Nilesh around telomere length and another biological biomarker for age, which is with methylomics. So DNA methylation has been associated with age as a biological biomarker. And Nilesh, I wonder, the question is, how much has the telomere length been compared to methylation as a biomarker for age?
Matt, I really don't have an answer to that question. I mean, there are different signatures for age. You're right that I don't give the impression that telomere is the only biomarker for age. There are a whole bunch of different things that people have looked at. I think as far as I can tell, there haven't been enormous scale studies that allow us to dissect that out in terms of methylation and the signatures that come from DNA methylation. I've seen the papers on DNA methylation, but those studies haven't really had telomere length measurements integrated as far as I'm aware to give us confidence. I suspect there will be a link of some sort, but I think they may, whether they're independent or joint, I really can't give you the answer now.
Thanks, Nilesh. Well, we look forward to hearing what the answer might be. And there was just a question, Tony, in response to your mention of the metabolomics data, there was a question about whether there's a release date for the metabolomics data.
I don't have a specific release date. I don't think there is a specific release date. It's just UK Biobank suggests that it is imminent.
Okay, thank you for that. And then there was a question relating to the choice of platform for the proteomics, Chris. And I wondered whether you could comment there's one of the queries is around the potential for using SomaLogic as an approach, which and what were the kind of the key decision points that kind of enabled you to settle on Olink?
Sure. Yeah, I was hoping that question wouldn't be asked because it's such a controversial topic, but I'm happy to answer it. First and foremost, both platforms are just excellent. They're extremely high quality technologies, and that's beyond question. And in some ways, it's a little bit like an apples and oranges comparison, but let me get into the weeds for a second. So when we initiated this effort, the discussion was mainly centered around Olink or SomaLogic. At the time, those were the two technologies we felt were capable of scaling up to UK Biobank's level. With SomaLogic, you can currently capture more proteins, up to 7,000. With Olink, in this year, it will go to 3,000. So obviously, it's less proteins, but there was a belief among some of the scientists that Olink's platform offers more specificity. It does a really, really good job of capturing the proteins it's supposed to. Soma does a great job too, but because it has more proteins, it arguably requires more downstream deconvolution of the signals for certain subsections of proteins. So it's a little bit of a question of whether you want breadth or depth. It reminds me in a way of Illumina versus AFI in the early days of gene sequencing. In this instance, we decided to move forward with depth and Olink, but ultimately it's a little bit like splitting hairs. I spent most of 2020 having this debate, and we eventually chose Olink in part because of the specificity and in part because of pragmatic issues. They seemed like they were going to be the easiest company to work with from a logistics standpoint. They're well regarded for their customer service. They offered an incredibly competitive price, but we still very much regard the SomaLogic platform very highly. We would eventually like to do some sort of type of head-to-head comparison between Soma and Olink, and potentially some of these even newer platforms emerging over the last 6 to 12 months. But yeah, I would hate to put it in terms of which platform is superior. They both do different things depending on what the question you want to answer and depending on the kind of data that you want to leverage.
Thanks, Chris. And one clarifying question, which I think many people might be interested in, with respect to the samples that are being used for the proteomics analysis. Are these the samples that were taken at baseline on the UK Biobank participants and therefore have the most prospective kind of opportunities?
Yes. So that was written on my slides, but I didn't get a chance to clarify that. So thanks for sending in that question. They will be the baseline samples, except for the COVID-19 component, those 6,000 from samples from the study that Paul Matthews mentioned. But apart from that, these will all be baseline collected approximately 10 years ago.
Mm-hmm. Mm-hmm. Great, and then shifting gears somewhat, Nilesh, lots of questions about telomeres. One around the genetic data that you presented with that over 100 novel loci for associating with telomere length. One of the questions was what proportion of the variance in telomere length is explained now by this kind of much more granular genetic picture?
Yeah, it's we calculate about 4.7%, which is typical for common variants, that it doesn't explain, the vast majority is about 4.7% that we explain with the common variants. We've also got just to say, Matt, we also got a sort of, we applied a very stringent p-value, of course, to the data because of the size of the genetic pool that we were testing compared to most things. But we also got about 500 additional sort of genetic variants which fit within an FDR list of 1 to 1%. So there is a lot more genetic variants than the ones I've mentioned that we feel very most comfortable about. But the amount of variation explained is common for typical biomedical traits that these number of variants would explain from around 4 to 5%. So it provides a reasonable instrument for undertaking Mendelian randomization, but not extremely powerful one.
Mm-hmm. That's really helpful, Nilesh. And then another question on the topic of telomeres, which I think brings into play the power of kind of incorporating information on the relatives of biobank participants. And the question is telomere length associated with the longevity of the participants' parents?
What a great question, and one that's been around for a while as well. So, we had the opportunity, of course, of looking at or modeling longevity of the people in UK Biobank themselves, but we obviously looked. The proportion of the UK Biobank participants told us about their parental history and how long they lived for. So So, and we do see associations between both paternal and maternal longevity. At least in the observation analysis, the genetic analyses are not as conclusive. So we don't know whether there's a causal relationship, but we do see relations between maternal and paternal age at death, if I can put it like that, or longevity. Yeah. So additional support in a sense that it does have some role in your life expectancy.
Thank you for that, Nilesh. And then lots of interest in the proteomics and some of the details around the data. And one of the questions, Chris, is whether the list of proteins that are being assayed is available.
Yes, that's an easy one to answer. If you go to olink.com, it should be very easy to download the full Excel spreadsheet containing the UniProt IDs and believe the Hugo gene names as well.
Mm-hmm. And then a second one was about the samples that are being cherry-picked. I mean, obviously, other than the COVID-related ones, how were they selected, and how maybe do they overlap with some of the other enhanced phenotyping or omic data sets?
Yeah, great question. I mean, the 5,000 cherry-picked samples were designed so that, it will give each of the 10 companies involved an incentive to invest in this financially. So let's just say in the example of Biogen, everyone knows that Biogen is very interested in Alzheimer's disease. And so we may, if we only had randomized samples, there's no guarantee that we would capture as many Alzheimer's disease cases as possible. So that gave us the opportunity to basically tell UKB, listen, we have this sample of 5,000. Can you make sure you include as many Alzheimer's cases as possible? Or in the case of Pfizer, maybe they'll want somebody with a particularly high polygenic risk score for a disease they're very interested in treating. Or maybe they want to capture everyone who has undergone MRI scans. So that basically, we basically hedged our bets, 45,000 randomized in order to keep things as representative as possible of the general population, but a smaller 5,000 subset, just to make sure that when we do get these data, there will be some phenotypes that we're interested in and that we have investments in treating.
And maybe if I just take chair's privilege on one particular question. So Cecilia, you mentioned about the genetic data and you mentioned the ACMG59 genes associated with late onset disorders. And I wondered whether you could comment on whether we've learned much about the penetrance of some of these genetic variants from studying UK Biobank that maybe we hadn't appreciated from studying patient cohorts in the past.
So I think that's a really good question. I don't think we've sort of interrogated that fully yet. I do think that the odds ratios would indicate, I think the median odds ratio is around 15, where the median odds ratio for any one common variant is, a 15th of that roughly. So, or a little bit more, but still. So I think that if odds ratio is any sort of correlation with penetrance, then yes, we have seen that they're higher. But for me, penetrance needs to be demonstrated in a physiological system afterwards to fully appreciate, because odds ratios is a statistical value and they need to be interrogated in the proper sort of model system to be fully evaluated. I don't know if you agree, but I would want to study it functionally then and optimally, maybe in a recall by genotype experiment where you would properly sort of investigate it.
Mm. Yes, I think there's a few studies that have started getting into this space using the biobank data, and you cited a couple on your slides from Caroline Wright, suggesting that the penetrance is actually much lower than sometimes we might think from disease studies, and the value of combining disease studies and population studies, I think, is really powerful. Will only continue.
And Caroline also pointed out the caution we need to have around the QC and sorry, I just wanted to put that in. So therefore, I think that functional validation and then also physiological validation is going to be key to really fully extrapolate that. That's why I hesitated a bit around it.
Yeah, no, that's a very good point. Now, so maybe one final question to draw this panel to a close, and I think this may be a question from someone who's feeling they might have short telomeres, but the question is, what kind of behaviours, Nilesh, might protect against some of the deleterious effects of telomere length? And is there ways of using the Biobank data to get into that?
I think absolutely. I think UK Biobank provides us with an enormous opportunity to look at this, Matt, in detail. There have been lots of suggestions in the past, about omega-3 fatty acid intake and telomere length, particular sorts of diets. So I think, this data provides a really fantastic means of exploring it. And if I can just finish with two sort of other sort of just general points. Of course, with the follow-up measurements, one of the benefits might be that in due course, we repeat those telomere measurements on follow-up DNA samples, which will allow us to have a handle on telomere attrition rates. And therefore, you can, rather than just a static measurements at baseline, you'll be able to get that. And finally, I think lots of colleagues today have thanked the UK Biobank team for this, for working with us. And I need to just express the same thanks, really, to Rory and his colleagues at UK Biobank in helping the telomere project as well.
Wonderful. A perfect note to end on. Thank you very much, Nilesh. And so just to draw this session to a close, I'd like to thank all of the speakers and the panelists and all of you for the great questions that came in. And I'm sorry we couldn't actually address all of them, but I hope you all enjoyed this session. So now we move on to the closing remarks, and it's a great pleasure to introduce the Right Honourable Professor, the Lord Kekar, who comes here primarily as the chair of UK Biobank, but in his own right is a professor of surgery at University College London, is chair of the UCL Partners Academic Health Science Partnership, and director of the Thrombosis Research Institute. And he also serves in one of his many other roles as the chairman of the King's Fund. And so it's a great pleasure to introduce you, Lord Kekar, and I'd like to hand over to you for the closing remarks.
Well, thank you very much indeed. I'm most grateful. And thank you to, first of all those who've contributed to today's remarkable symposium. Really absolutely fascinating. But as chairman of the Board of Trustees of UK Biobank, which is the reason I'm here today, I would like to say to the global research community how grateful the board are for the collaboration, commitment, and opportunity to work with such a remarkable group of scientists globally. Some 19,000 researchers, as we have heard, undertaking some 2,500 approved applications and projects, with nearly 2,000 publications and, quite remarkably, 38,000 citations. That is really a remarkable biomedical collaboration, and something that all those who have decided to commit to this initiative and endeavour should be very proud of. What's very clear in the board, and in fact amongst funders and others, is the fact that although this has been a long journey, some 20 years for Biobank so far, we're really only in the foothills of what is, I think, a remarkable opportunity to contribute to scientific understanding and to mankind, and to the understanding of disease, and indeed the improvement of human health. There has been, of course, substantial external investment, and as we've seen today, some of the fruits of that investment in terms of the application of new assays and technologies, and how all of those data are going to provide even more and more resource in the years to come, as we're able to apply more to the Biobank resource. But in addition to that, in addition to all the assay data, we're also in the fortunate position of having more and more clinical follow-up data. And that is, of course, critically important and adds to the unique value of this resource, something that's recognized, of course, globally. And therefore, I and the board see the opportunity for Biobank to be even greater in the next 5 to 10 years. And we need to all mobilize together as a global scientific community and ensure that we make full use of the trust and contribution of the participants, those who gave their biological samples and have given their permission for us to collect follow-up data on them. Because that trust, given some 20 years ago, was a great gift to science and to understanding, and something that needs to be marshalled and used in the most appropriate fashion to answer critical questions as we advance our capacity, our knowledge, our scientific skills to do so. Now, what's been very interesting as we start to look at the way that the resource is being used is, to date, much of the utilization has been from higher income countries, from, of course, the United Kingdom, United States, Canada, Australia, and Europe, and so on. But with the establishment now of our research analysis platform that's going to become available to all researchers later in the year, and with the added benefit, of course, of free computational time for researchers from low and middle income countries, and for students and for early career researchers, I very much hope that this resource is going to be made more broadly available. So those with real imagination, those with a sense to wish to explore, to apply their intellectual ability and their scientific and clinical knowledge, can access this resource. There is a deep commitment in Biobank to democratizing research as best we can, to assisting the entire global research community, to ensure that what we believe to be a unique resource can be applied throughout the world. And in so doing, and in sharing the results of that endeavor, we're going to be able, I think, to accelerate the way that we can address and answer critical questions and move forward the opportunities, as I've said, to better understand and characterize disease, but also to identify new therapeutic targets and to validate associations and relationships that only a program like this, as we've heard in this last session, is able to do. Really, as I've said, my duty is to thank everybody. So, of course, to thank our funders. Without that deep commitment to the continued funding of this program, we wouldn't be able to continue. And I know that there is a deep commitment to continue to fund this important resource. Of course, to all the scientist collaborators from around the world, it is their ability to identify questions, to apply their skills, their intellect, that provides the tremendous results through evaluation and exploitation of the resource that we have. The remarkable Biobank team led by Rory Collins, a remarkable scientist who's deeply committed not only to all the research, but to the values that drive our commitment to a global effort open to all to participate. And of course, I should mark and congratulate him on being the recipient of the MRC Millennium Medal for 2020, a very important recognition of his tremendous contributions, not only to Biobank, but more broadly in science. But then most of all, a very deep appreciation and gratitude to the Biobank participants. As I've said, it's their commitment that has made all of this possible. So as we move forward, the board are committed to using every opportunity to grow what can be delivered through Biobank, and we look forward to doing that with what I consider to be a global community participating in one of the most exciting and impressive global biomedical research projects. Thank you all very much indeed.